I would like to start by thanking Virginie and Abdu for putting together this fantastic conference. I know we're in the last section now, so I just want to take a moment and celebrate how great this has been to get everyone together. I also wanted to say that I took to heart your guidance to kind of look for work that puts together as much as we can ecology and evolution, which is exciting because it gives me a chance to talk about some things we've done in the last couple of years, but framed by work that started long ago. So it's kind of a mix of some new and some old here today. I also think that it is fitting, given Amir's beautiful tribute to Jean David at the open of the conference, to be celebrating Drosophila pigmentation today. In fact, I want to start by trying to share sort of why this is such a great system with some of you, but I feel a little self-conscious because there's probably 5 to 10, I think I've counted, of you in this audience that also have worked in this system. So you can tone off for a minute, and the rest of you, I'll try to convince you why it's a great system. So after I do that, I'm going to focus on one specific case study today, a pair of closely related species, and tell you about the genetic basis of pigmentation differences in body color between them, look at variation within one of the species, and close by some hand-wavy comments related to possible selection pressures that may be driving the evolution of pigmentation in this group. So one of the key features, if you're going to study evolutionary diversity, is to have diversity, right? And we have plenty of that when it comes to Drosophila pigmentation. We might not have the pretty colors that butterflies or some of the other organisms we've seen talks on in the last 2 days have, but we do have great variety in space, sort of spots and stripes and shades of yellow, brown, and tan pigments. We also see a lot of variation within a species. I've pulled an example here from Eloise and Amir, sort of highlighting some of the big pigmentation diversity that exists within Drosophila melanogaster, the best studied Drosophila species. I'm guessing this was at least in part inspired perhaps by some of Jean David's sampling in Africa. I'll also note that in this paper, they report a correlation between body color and UV radiation. And I just want to point out that differences in pigmentation among Drosophila species have been kind of implicated in a variety of sort of ecological processes yeah, I guess I'll stick with that. So camouflage, thermoregulation, desiccation resistance, resistance to parasite infections, protection from UV radiation, and then also courtship behavior and mate choice. In most of the cases, what you find is correlations between pigmentation and some factors that suggest that these are the selective forces. It's very, very difficult to really kind of put a causal link there, and I will be also unlikely to do that at the end of today. But I'll show you some suggestive data. Okay, Drosophila pigmentation is really determined developmentally during the pupal stages. We do have a pointer, it seems, with a button. Okay, so the Drosophila life cycle, right, lays an embryo, 3 larval stages, it pupates, and it's during these late pupal stages that the developmental processes, the spatial controlled gene expression that's necessary to determine the adult pigment patterns is really laid down. So in flies, the pigments are actually part of the adult cuticle. They're really incorporated within it. So the flies take up tyrosine from their diet through biochemical reactions that happen primarily in the epidermal cells and export into the developing cuticle. We get the production of different pigments. One of the great things about studying Drosophila pigmentation is we know a lot about the biochemistry and the genetic control of these processes. So here's the core biochemical pathway, and I just want to focus on a couple things. The first is I've kind of positioned this pathway to put dopamine at the middle, mostly because it highlights that we've got kind of branches coming off of it. So it's kind of a branched pathway to produce black, brown, yellow, and a colorless pigment, depending on sort of the amount and presence of different enzymes. So those enzymes, we know which genes encode them. They're now shown here in red. And I want to take a brief moment to point out the naming convention in Drosophila because this can get a little confusing at times. I'll use the yellow gene as an example. So the yellow gene, actually here's the mutant phenotype. So this is the adult abdominal cuticle in a wild type Drosophila melanogaster. This is a yellow mutant, a tan mutant, and an ebony mutant. The yellow gene was given that name by Thomas Hunt Morgan over 100 years ago because the fly looked yellow. What this means, though, is that the mutation that breaks this gene's function is what makes the fly look yellow, and its normal function is to make black. So yellow makes black, ebony makes yellow, and tan's kind of in the middle, okay? So just, it's the opposite of what it sounds like. All right, the other thing that we've learned from studying Drosophila development is that the spatial control of expression of these enzymes, particularly yellow, ebony, and tan, determines the spatial pattern of pigments. So I'm going to show you an example here focusing on ebony and tan, and I'm going to highlight that ebony and tan actually encode two enzymes that catalyze a reversible biochemical reaction. And that reaction is sitting in that branched pathway as sort of a switch between a more yellow pigment and a darker kind of tan, brownish pigment. So when we use the tools we have available in Drosophila melanogaster to turn these genes on where they might not normally be or at higher levels than they normally are, so I'm going to, in the next set of slides that I'll bring up here, the images, we'll be turning on expression in this kind of midline here. So there's a wild type abdomen. If we turn on ebony expression, we change that pigmentation from the wild type to a yellow stripe. If we turn on tan in that region instead of ebony, we create more of the dark pigments. And if we turn them on both together, we get something in between, so sort of capturing that balance that's happening in the biochemical pathway. Okay. So this is what happens developmentally, and this is what we can do in the lab by manipulating gene expression. There's a body of work that I will not review today, including from many in this room, showing that changes in expression often correlate with changes in the spatial pattern of pigmentation among Drosophila species. We'll see hints of that, but that won't be the focus of my talk today. So as I said, I'm going to focus on one species pair, a closely related species pair that differs in pigmentation, but is also able to produce fertile F1 hybrids that allow us to bring genetics to the table. So the species are members of the Virulus group. The Virulus group is diverged about 60 million years, depending on which paper you read, from the melanogaster group of Drosophila. And what's notable is this species, Drosophila nova mexicana, that has evolved a derived yellow body color in contrast to the darker body color of all the other members of this species group. Drosophila nova mexicana and americana last shared a common ancestor less than 500,000 years ago. And again, they can mate in the lab, make fertile hybrids. We can do back crosses and so on to do genetics. And if you were staring at those, these flies have no heads. Those of you that might be, just in case you're wondering where that third segment went. Okay, so as I mentioned, pigmentation is really happening during the late pupal stages, and you can see that pigmentation difference that we see in the adult arising even earlier in development. When we look at expression of both tan and ebony at these earlier developmental timescales, we do in fact see an expression difference, although for tan it's small. It's about a 1.2-fold difference. If we had done an RNA-seq experiment, that's probably not something we'd be excited about, but we can see it reliably. It's statistically significant, and you'll see meaningful. So what we see is that the darker species, Americana, has higher levels of, as I'm saying this out loud, I think I switched my coloring here, has higher levels of tan, and this is the gene that promotes those darker pigments than Nova mexicana, whereas Nova mexicana has higher levels of ebony, which promotes the yellow color. And yes, I switched my colors here, apologies. Okay, so we do see an expression difference between these two species. The genetic changes responsible for that may be at those genes, or they could be somewhere else in the genome. And to sort that out, we really need to do genetics, right? We need some way to find the actual genetic change causing this pigmentation difference. So this is where the genetics come to the table. We cross the two species, Americana, Nova mexicana, and make F1 hybrids. These are my schematics of an X chromosome and sort of a generic autosome, sort of lumping them all together here. So the F1 hybrid, you can see that dark pigmentation is largely dominant. We then backcross the F1 hybrid to Nova mexicana, the yellow species, and get a set of backcross progeny that are each a unique combination of the two parental alleles, the Americana and Nova mexicana alleles, and then have one full set of alleles from the yellow species. When I look at that distribution of phenotypes, it is not continuous, so that's point one. It actually falls into 5 discrete classes, and we've done this experiment many times over, embarrassingly, 20 years, all the way back to my postdoc. Many people have looked at this, and I've said, what do you see? And they see 5 classes. These 5 classes are illustrated here, ranged from light to dark, and this already starts to tell us something about the genetic architecture underlying this trait, right? It's not highly polygenic, the fact that we see these discrete classes. Back in the day, before you could easily sequence a whole genome, we still did QTL mapping. And so this is an early day kind of QTL map. And the only thing I want you to take from it is we do see genetic associations between the regions of the genome containing tan and ebony with that phenotype. Okay, so we think those two regions of the genome are contributing to the pigmentation difference, although you might note that the other markers linked to ebony are also highly correlated with the trait. I'll come back to that. So for tan, we were able to continue to use this sort of power of genetics and do fine scale mapping. And after searching through 10,000 flies, we found 2 recombinants with breakpoints in the middle of the tan gene that allow us to see that when you have basically this first intron of tan from Nova mexicana, the yellow species, or Americana, the brown species, you have lighter or darker pigmentation. So this is telling us there's some functional difference in this region. We were interested in mapping this further, so we decided to switch from a sort of genetic mapping approach and move to a transgenic approach by cloning the entire tan gene, regulatory region, and coding sequence from Drosophila nova mexicana and Americana and putting them into Drosophila nova mexicana. So this is the sort of dissected abdominal cuticle from Drosophila nova mexicana, the yellow species. When we put in the nova mexicana allele, it darkens pigmentation a little bit, right? This is, again, an extra copy. And when we put in the Americana allele, it darkens pigmentation much more. So again, we're confirming functionally that there are something different between these two alleles that's affecting pigmentation. We then spent a couple students and many years trying to map exactly what that is in the first intron. And I wanted to include this because I think we always show the things that worked out great. And this is an example where we just hit a brick wall through a combination of both technical challenges and biological challenges. So for instance, there's epistatic interactions, there's multiple sites within that intron that have effects in opposing directions, and it gets really messy really quick. So that's where that sort of story ends. For ebony, though, we couldn't even get that far because ebony is looped up in this lovely inversion, which we've heard a couple nice talks about during the meeting. When you are trying to do genetic mapping, inversions are not your friend. You can't get recombinants within there. So for ebony, we were stuck. We were also stuck with transgenic approaches because ebony is just a really big gene when you get all of its regulatory sequences as well. So this is where we needed to wait for technology to move forward. Fortunately, it has, CRISPR in particular, and we've been able to more recently use a reciprocal hemizygosity mapping strategy that David Stern nicely reviewed in 2014. It works like this. So if you take, if you have a candidate gene, in this case we wanted to test whether ebony has divergent sequences in it between the two species that might affect pigmentation. And so we did so by mutating ebony in both of the species, crossing the ebony mutant to the other species in both cases, sort of both directions, such that we have 2 genetically identical flies that differ only for the presence of one ebony allele that comes either from Americana or from Nova mexicana. Okay. I want to first show you what those ebony mutants look like. So again, we did this with CRISPR. For Nova mexicana, the yellow species, an ebony mutant makes the fly look dark. It is a recessive allele, so it was sort of not trivial to find this, but it was somewhat easy to look for the darker flies. An ebony mutant in Americana, however, makes a dark fly darker. So this was a little harder to find, but we did. We had one clue, which was in Nova mexicana. We discovered the ebony mutants had a phenotype in the pupil case, which is actually what saved us here for Americana. So we have mutants now in both species. Thanks to CRISPR, we can do the hemizygous test and finally show, indeed, there is functional divergence at ebony that affects pigmentation, and those are the differences, right? Okay. Again, when you put this together, if we look at how much do ebony and tan versus other genes in the genome contribute to this phenotype, if we quantify sort of pigmentation color in these images, ebony and tan together explain about 87% of the difference. So it's largely these 2 genes explaining the difference between the 2 species. Okay. So then what's going on within species? So in Drosophila americana is found through most of the United States, kind of east of the Rocky Mountains, whereas Nova mexicana is found in the southwest. Drosophila americana has pigmentation variation within the species, and specifically a longitudinal cline for body color. So populations closer to the Rocky Mountains have lighter pigmentation, and as you move further east, it becomes darker. Okay. When we see a cline, there's a few things that we could suspect or we might want to rule out. So maybe there's some population structure going on here. Maybe there's sort of a dispersal, isolation by distance component, or maybe it's local adaptation, right? Differences in the environment across the range. A number of studies from others as well as our own group show that there's no evidence of population structure in the species, so we rule that out. We also didn't see evidence of isolation by distance based on patterns of genetic variation at different loci. So this suggests that maybe it is indeed local adaptation. That's what the data are pointing to. And what we wanted to know first was when we look at these different strains of Americana, some that have lighter pigmentation versus darker, are they using the same genes, tan and ebony in particular, that were responsible for that difference between species to sort of generate this variation? So in the first time we looked at this, we looked specifically at the lines shown here. The names for you are largely meaningless, but know that they come from different geographic locations. This dark line is the one that we used for all the crossing between species I've shown you so far. We took 3 lines from this one population in Nebraska and 1 line that also has light pigmentation, one other line from a different location. Okay, we use the same type of genetic analysis, cross, make the F1 hybrids, do the back cross to Nova Mexicana again. So in this case, we're not mapping between species. Well, we are, but that's not the goal. The goal is to really compare between the different strains, and we're using Nova Mexicana as a common reference point in the experiment. The first thing that we can see just from looking at those back cross progeny, and as a geneticist, I just kind of love this, right? It's a reminder of how much we can actually infer about genetic architecture from just phenotypes, right, without ever sequencing anything. So what we see is different strains that came in one case from the same population. Those back cross populations produce different colors and different frequencies of those colors of flies. So that's already telling us there's something different about the genetic architecture. We then went to test specifically for divergence of ebony, tan, and we included yellow, which was not contributing to the interspecific divergence as a control. So this is the data I showed you already in the first part of the talk, summarized in this table where A represents the Americana allele and the Nova Mexicana sort of function of that allele. One of the first light strains we looked at also showed divergence of both ebony and tan between Americana and this lighter between Nova Mexicana and the lighter strain of Americana. So there's something else presumably making that lighter. When we got to this next strain, we were a little bit surprised to see that actually there was no functional difference for ebony between these lines. It actually turns out that this has a functionally equivalent allele that shares some sequence similarity and even is wrapped up in that inversion as well. So it was actually segregating in Americana, it seems, prior to the fixation in Nova Mexicana. The next line we looked at had the opposite surprise, which was this one had an allele of tan that was functionally equivalent and shared some sequence similarity to the Nova Mexicana allele. So what we have here already is we can see we have 3 different ways of generating this light pigmentation. Right? All right, so kind of putting that together, some of the genetics with the biochemistry and development that I told you about earlier gives us an understanding that looks like this. So if we have flies, and this is from one back cross population, we have some individuals that inherited the Nova Mexicana allele of both ebony and tan, and when they do, they tend to have very light pigmentation. If they inherit both the Americana allele of ebony and tan, they tend to have very dark pigmentation. And if they get one of each, in either combination, they tend to have this intermediate sort of lighter pigmentation. Now, I showed you already that ebony is expressed at higher levels in Nova Mexicana, and tan is expressed at higher levels in Americana. And so when we put these alleles together, we have this one driving towards the yellow, creating yellow body color, this one creating a brown body color. And then in these intermediate groups, we have different combinations of alleles. This one has both the higher expressing alleles of tan and ebony, this the lower expressing tan and alleles of tan and ebony, but what's key is they're balanced in both cases, and so you get these intermediate phenotypes. In fact, that's what we saw in the DN2 strain and the A01 strains. Okay, so there's multiple ways to make a similarly light pigmentation. We then wanted to look more carefully at the full range of pigmentation in Drosophila americana. So again, we have this kind of nationwide cline, if you will. And so we wanted to say, okay, well, what is the frequency of those Nova Mexicana-like alleles throughout this range? So often when people are trying to understand genes that might be contributing to clinal variation, and you start with a candidate gene, you might look for a cline in allele frequency of that gene. So we wanted to look at how often those Nova Mexicana and, the tan and ebony alleles that were like Nova Mexicana were found in these Americana populations. We did this by sequencing because we had that sequence similarity as well as the functional similarity. And what we learned is that we were super lucky the first time out because we didn't find any other alleles throughout the species range that we sampled that had that same kind of sequence and sequence similarity. So then we wanted to better understand the functional role that they might play, even if we can't pick them up by sequence. And we did this by repeating this backcross for 51 different isofemale lines. 1 to 5 had come from each of 20 different geographic populations. So we did the backcross, we analyzed the phenotypes in that backcross population, and genotyped for ebony and tan and yellow as a control. Again, the first thing we noticed was that different populations, so the MK, the first 2 letters are basically a designation of location, so these are all from 5 different locations. And what we saw was they're producing different distributions of phenotypes, again, underscoring that there's differences in genetic architecture in the populations. When we put all the data together, we were surprised to see actually there's a lot of variation in genetic architecture even within the same population. So this is a slightly complicated figure. It's basically a visual representation. I don't think I need to do that. Of the colors. So the lighter yellow represents the lighter classes, the darker classes. And maybe I can highlight this. I don't know what this is doing. Okay. We'll go all the way down here to this example. This is 2 different flies isolated from the same population in the same collection year, and one of them produces some of our lightest classes of flies, whereas the other in the same type of backcross produced many of the darker flies. So clearly we have different genetic architectures segregating the same population, so genetic heterogeneity, even though we have this cline, right? So we have this local adaptation pattern at a large scale, but when we look within an individual population, we have genetic variability, possibly because of multiple ways to create the same pigmentation phenotype. Okay. So now the slightly more speculative thing, what might be driving this cline or pigmentation evolution in this species in general? We tested for correlations between that clinal pigmentation and a number of environmental variables. The best correlation seemed to be with humidity, relative humidity, suggesting that maybe it has to do with desiccation resistance. So we tested this, and I'll say we tested this in a way that I think was a good way to test this. And what I mean by that is we took Nova Mexicana flies, and one of which we had introgressed Americana alleles. So the flies are almost genetically identical except for the alleles that affect pigmentation. And when we compared their desiccation resistance, measured in the way we do in the lab, which is pretty artificial, like put them in a vial with desiccant and count how long does it take them to die, right? Presumably that's not the selection pressure they have in the wild, but that's what Drosophila geneticists do. So we saw no significant difference between them. Nonetheless, more recently, Leone Morel's group has published a paper suggesting that there is possibly desiccation resistance that differs within Americana, that correlates with pigmentation, although I will point out that these are sort of the whole flies being compared, not that kind of small difference that I pointed out. So, but to me it puts, okay, maybe desiccation resistance back on the table. This paper, even more recent, caught my eye again because it links desiccation resistance to patterns of cuticular hydrocarbons. So these are molecules that the flies produce on their abdomens. And the reason it caught my eye was that a couple years before that paper came out, we had shown that actually tan and ebony mutants change cuticular hydrocarbon length. So they shift the distribution. One of them, so let's see, ebony shifts towards, so an ebony mutant shifts towards longer chain CHCs, whereas a tan mutant, again, remember these are opposing biochemical functions, shifts towards shorter chain CHCs. There's also a correlation between CHC length and abdominal pigmentation in the DGRP line of or population of Drosophila. Okay. So it suggests there may be variation linked between them in the wild too. We also used our ebony mutants to look for differences in cuticular hydrocarbon length. And what we found was that when we knock out ebony in these two species, it affects it affects CHC length in both cases, and does so in opposing ways, which we think has to do with the different tan activities that we know are present in these flies. This becomes even more intriguing to me when, again, from Leonie Moyle's group, they find that a shift to these shorter cuticular hydrocarbons accompanies sexual isolation among different populations in this species. So now we have the case where these pigmentation genes don't just affect pigmentation, they also affect CHCs. This may affect desiccation, it may affect mating behavior, and so on. So pleiotropy may be sort of complicating interpretations here. And then at the coffee break today, here, I learned about Jean-Michel's recent work where he has tested a thermal kind of tolerance hypothesis by shining light on Americana and Nova Mexicana and measuring their body temperature under the same conditions. And in fact, was able to demonstrate that Americana does get warmer, like its body temperature goes up in that same amount of light relative to Nova Mexicana. And I'll remind you that this is the one found in the warmer, like desert climates, whereas this is more in the cooler climates. All right, so with that, I hope you leave with three things. The first is that tan and ebony, I think, are primarily responsible for the divergence between species, but identifying specific nucleotide changes has been very challenging, and that's based on a decade plus of work trying to do so. And I think that's extrapolatable to other traits in other species. Local adaptation for body color in Americana, again, we see this broad cline, but a lot of complexity with multiple genetic mechanisms, even within a population, which suggests to me that had we just scanned for an allele frequency cline, as we actually did, right, we wouldn't see anything because of that complexity. And finally, in terms of possible selection pressures, these changes in CHCs, which may in fact cause changes in desiccation resistance, might be contributing to that cline. But disentangling whether that's CHCs or actual color part of pigmentation is, again, I think, complicated by pleiotropy. So I want to thank the members of my group that did the work, sort of most recently included Abby Lamb, John Massey, and Ariel Cooley. And with that, thank you for your attention.